Alrighty. I believe that Harald Schwalbe is on the call.
Yes, I am.
Okay, perfect. Thank you for joining us, Harald. So our first speaker today is Dr. Harald Schwalbe, and he is the professor at the Johann Wolfgang Goethe University in Germany. Harald Schwalbe obtained his PhD in chemistry in 1995 from Goethe University under the supervision of Professor Christian Gresslinger. His graduate research was on NMR method development and protein NMR spectroscopy. He was postdoctoral fellow with Professor Sir Chris Dobson, FRS, at Oxford University. From 1995 to 1999, he worked on his habilitation. He accepted an offer to become assistant professor at the Massachusetts Institute for Technology in 1999. In 2002, he became a full professor of organic chemistry at Goethe University, and his research focuses on the investigation of structures and dynamics of proteins, DNA, and RNA, and their complexes, particularly with NMR spectroscopy. So today he's going to go through and give us a little bit more information about this talk that he has entitled Rapid RNA NMR Assignment Process. So without further ado, Harald, I'm going to stop sharing my screen. And if you can share your screen, the floor will be yours.
I hope you see my presentation.
Yes, we can see it.
Okay, I'm happy to be here. These are short presentations, and I like to focus on RNA and give you first a flavor of what we mean by being able to contribute by NMR in something relevant, and that's of course SARS-CoV-2 nowadays. And in March 2020, we thought about the fact that a virus compared to a human cell is small. SARS-CoV for a virus is large, but for a human cell it's small. And there are only 27 proteins in the proteome and only 30,000 nucleotides in the RNA genome. And if we pull forces together, we should actually be able to look at all of these elements and their domains, the small, and we established a network where now 260 people actually worked. And the aim of this network was to assign NMR spectra and to determine where missing RNA and protein structures, and then conduct ligand screening campaigns to help identifying pharmacologically relevant binders. And so one of the things is proteins, and here this was published in 2021, a set of 25 of those 27, 29 proteins and NMR, and you see the HSQC fingerprints. So once we have this HSQC, we are in business. We can start characterizing these isolated domains. And the advantage of NMR is that we can look also at intrinsically disordered proteins. We can look at potential drug targets. And when we do this holistically and not in isolation, then we can identify targets that have not yet been on the radar screen before. But I would like to focus on RNA, and basically what I show to you here comes from a 2020 publication by Anna Wacker and Julia Weigand from our consortium in Frankfurt and Darmstadt. And you see here that this is the 5' untranslated region of the virus. And then this is the 3' untranslated region of the virus. Sorry, I go back and try to activate my laser. And then in the middle, there's another interesting part, which is the pseudoknot. And down here, you see, for example, in isolation, this 5' SL1 comes from here, 5' SL2, 3, and so forth and so forth. Then you see here the entire RNA from end to up here. And then you see the entire full-length RNA from here to here. And each in this spectrum needs isotope labeling. And so we actually prepare multiple samples to actually be able to assign these spectra. And once we have assigned the spectra, we also want to contribute to the structural biology of this virus. And basically our aim is to determine each and every structure of each and every element of the virus. And their NMR is unique and is unmatched by X-ray, because X-ray can't solve RNA up to now so well. And one of the things that's also important is to communicate these results rapidly in an open sharing modus. So we're not conducting closed lab. Protein, it's certainly good that you have the assignments ready and you know how to express the protein, because that gives you a head start on conducting your own science. I give you one example that is worked by Clara Mertinkus on this element, which is the 5th stem loop element on the 5' side of the virus. It contains here the start codon for the open reading frames A1, 1A and B. And you see that this is in total 149 nucleotides. It contains a 4-way junction. And we break this down into individual parts in red, 5SL, 5A, BC, then stem, and then the junction. And when you do that, here you see the HSQC spectra of these individual elements as indicated in colors. Then you see that the sum of all overlaps with the individual elements, making such divide and conquer approach feasible. And you also see basically the SAX envelope. This is work conducted by Andreas Schlund from Frankfurt, overlapped with models of these individual elements. And here I show to you this network or underground map of the experiments that we conduct to actually assign each and every nucleotide. And we typically prepare 15N labeled samples first, and then 15N 13C labeled samples, and then conduct these experiments. And basically, this is then for one RNA, it's kind of 14 days pure measurement to get these set of experiments in place and to get at least nucleobase assignment ready. And I just show to you the beauty of the spectrum by way of a small element. It's 5SL, 5B. We start with the TROSY, count the number of non-exchangeable amino protons, connect those by NOSY experiments, conduct HNN COSY experiments that tell us something about the hydrogen bond status. And then we use also the HSQC, the NOSY, and the carbon-13 HSQC to identify connectivities. And then from that goes further from the nucleobases to the carbons. And I'd like to point to a experiment here that is a proton-proton X filtered NOSY, where we have partial labeling, and so we can filter out those nucleotides that are not labeled. That helps simplifying the spectrum by a spectroscopic manner, and then we go about and assign our spectra. This is also here an HCCH toxi with various mixing times to get connectivities for 1, 2. This is a typo here, it should be 1, 2, and up to the carbon 5 in the sugar. And with these assignments, just to show you here one immediate example, this is wild type sequence 5SL5B. And this is a mutant in the variants of concern that actually accumulates. So this C at 241 is replaced by U. This then becomes a dominant mutation in Omicron. And so then we can immediately repeat this process. And since little has changed, here is a changing thing, and so forth, here are changes, then we can immediately assign this. So we are ready, once a new variant of concern shows up, to actually see whether the structure is identical or has changed. And for all of this, we need isotope labeling. I'd like to also now add one more in the last part of a new method to introduce a single nucleotide into a large RNA. And the current methodologies that also we heard just a second ago is often you use in vitro transcription. And then you have a DNA that is read off by a polymerase, and then you label this RNA strand. And of course, you can add all 4 nucleotides 15N or all 4 nucleotides 15N 13C, or you can add, for example, uridine, only uridine 15N 13C, but then all uridines are labeled, so you get by in vitro transcription no site specificity. You can easily do that by solid phase synthesis, where, and these, as we heard, are also commercially available. You use phosphoramidites as building blocks. And of course, by choice of what phosphoramidite you use, you can incorporate a single nucleotide in whatever labeled form. And the group around Kreutz in Innsbruck is very active in also synthesizing new labels, and also Gerhard Wagner has very nice new labels out there. Kwaku Dai also uses new labels and so forth. So this is very important. But the problem with solid phase synthesis is that it has size limitation. I mean, this is already gracious to say you could make an 18 nucleotide long RNA by solid phase synthesis. In our hands, often even 50 nucleotide long RNAs are just not pure and cannot be used for NMR. So down here in 2018, and I will show you more about this in a minute, we have introduced a new method, and that works as follows. You take a modified nucleoside, and I'll show you the modification in a second, and then you add this chemical reagent, phosphoryl chloride, and that bisphosphorylates the 3' and the 5' side of the nucleoside. As byproduct, you also get the 2', 5' bisphosphorylated side, but this 2', 5' phosphorylated by nucleoside is not further incorporated. So you add this bisphosphate to an RNA and add an RNA ligase, and that then adds a single nucleotide, this bisphosphate. So then you get the RNA extended by 1 nucleotide, and as byproduct, the unreacted RNA that is the same as the starting material. Then you can, if need be, actually get rid of this unreacted starting material by oxidizing this with sodium periodate. And basically you have a D-keto group that you can also then remove. And then you take this wanted product and dephosphorylate at the 3' OH. So you get then here a phosphate, and then you can take a second RNA and do a splinted DNA ligation. So then you have an RNA that is modified at a single site. And now, initially, we showed that this would work for a number of, and this is work by Sarah Kehani-Goldau, and we, for example, also incorporated pseudouridine and photolabeled pseudouridine, or here we attached in a ribo switch this cytosine with a photolabel protecting group. And when the photolabel protecting group is on, then this C cannot bind its natural ligand desoxyguanosine. But when we irradiate with light, then it binds. And so we could use, so this ligase actually accepts a lot of substrates, and many of these with photolabel groups. And I just show you here again. So we have this label, and this is the paper up here, this label, and then we shine light, and then this C is free, and then it can bind its ligand. And so what you see here is the RNA, with the photolabel protecting group and the ligand, no binding, and then we shine light and we see the reporter signals for binding. This is on the side of the RNA. And when you take 15N labeled ligand and unlabeled RNA, you also see that up here comes the signal. Actually, it's 2 because there are 2 different binding modes of the ligand that binds to the ribose switch. And we could also add azobenzene at this final position of helix 1. And then in the E configuration of this azobenzene, ligand was bound. And in the Z configuration, it was not bound. So you see basically 45% reduction upon shining light on this azo-derivatized ribose switch. Now, there are 2 other aspects, and that is now unpublished work, but we soon submit by Bojana Knesic and Alexei Sudakov. And we want to see how far can we extend this procedure. And so we took natural occurring modified nucleotides, how they occur in tRNAs, ribosomal RNAs, and mRNAs. Here is FMET shown for E. coli. And here this U is a thiouridine, this uridine is a dehydrouridine, and so forth and so forth. And we tested whether these natural occurring modified nucleotides can also be incorporated by this method. And we also tested whether we could put pseudouridine under photolabel protection. And then in order to go further next to NMR, we also purchased fluorinated modified nucleosides. They are not natural, they are non-natural occurring modified nucleotides, but they are, of course, extremely good RNA NMR spies, as we know. And so we looked at whether we could incorporate those fluoro modifications, and we also looked at methyl modifications, because you can also think about 13C or the 13C deuterated reporters down here. And this all went into a big table that shows you as the take-home message that all of these can be incorporated. And mind you, what you have to do to make those commercial available precursors into something that you can incorporate in an RNA synthesis is just to add this phosphoryl chloride reagent in a one-step procedure. And then you see the bisphosphorylation yields are up here, but you really, the availability of this is not an issue. And so you can use this in unoptimized reaction conditions and get this bisphosphate. And when you do that, then we incorporated here at this C position a fluor and tested then. And then you see the beauty of fluor NMR, because here you see the proton 1D, and in the fluor 1D you see that actually the apostate are 2 states. So there is conformational heterogeneity in the apostate that you can positively detect by NMR, which you can by fluor NMR as opposed to proton NMR, and you see that this state APO2 goes down, APO1 goes down, and the holostate that is in the middle here actually goes up. So basically fluor NMR shows to you and allows you to detect conformational heterogeneity in these large RNAs. And with this, I'm done. I thank you for your attention, and I'm very happy if you could ask questions. And this is where we did our group trip. It's in Austria, it's in Achensee. So if you want to join my group, that would be great. And then we will take you to our group trip next time. Thank you very much.
Awesome. Thank you very much, Harald. We do have a couple questions in the Q&A box. First one is, you mentioned 14 days of NMR time. Can you provide a time estimate for the full process, including sample prep, NMR time, assignment, and structure calculation?
What I'm saying are optimized timelines that say people work on this that are experts. And over COVID-19, under separation policies of using the lab, we had teams that prepared 4 RNAs in HPLC purified quantity over 14 days. And so basically it takes you 14 days to make 1 RNA. Before that, you have to make the gene. We often order the gene, so it's so cheap nowadays that you might not want to do too many mutations yourself. It's just not time effective. From this, the 15N assignment and nucleobase assignment requires 14 days of measurement time, and if you are experienced and things work well, it's probably 2 weeks or 4 weeks for the assignment. Structure determination by NMR of RNA, I would not dare to predict. Sometimes it's fast, sometimes it's slow. It really depends. This is difficult to predict.
Wonderful. Thank you, Harald. And the second question that we have is, which strain or strains of COVID were studied?
We started with wild type when it came out and did then relevant mutations. In the RNA, the mutations are rare because the virus in the untranslated regions has not yet been challenged by anything. So all vaccination works on spike, and spike is coded. So the mutations are outside the area that really interests us as by the design of our study. And one should also say that the untranslated regions, as you go from MERS, SARS-CoV-1, but even down to alpha coronaviruses, are not very much affected up to now.
Wonderful. And I think we do have time for 2 very quick more questions. So regarding your last spectra showing 19F NMR, what field strength was used? Is there an optimal field strength for 19F NMR of large RNA, considering CSA effects?
I think probably Gerhard is better prepared to really answer this. But my 2 or 3 cents to this, we don't have fluor probes on all spectra as we have probes on proton, carbon, nitrogen on all spectra. So this is the one point. Second point, the prediction of the CSA of fluor is very difficult and it changes a lot. So simulations of these kind of things with a single fluor is complicated. Gerhard has a very nice thing with a fluor attached to cytosine, where a Rosie effect kicks in, and then he probably can answer better than I do. And he's raising his hand, Gerhard.
Yeah, I think, can I answer the question?
Yep,
we find
feel free to chime in.
600 megahertz is best for fluor NMR. So we have upgraded our instrument. We have a nice quintuple resonance fluor probe on a 600 megahertz instrument, and this seems to be the best field strength for fluor NMR.
Wonderful, thank you. And then one final question that we had for you is, what are the typical yields of your labeling strategy, and how does this compare to other methods such as PLOR?
What is PLOR?
Yes, such as PLOR.
Well, wouldn't know the abbreviation there at this point, but maybe it can be repeated in the chat. But for the ligation to the large RNAs, we can reach something like a 200 micromolar, 100-200 micromolar sample, given time restraints of PhD thesis and so forth. So it is not yet where we would love to have it. So if you do T7 polymerase reaction, you get millimolar samples, and that most of the time works well, although you have to optimize every new triphosphate mix that you get from CIL anew. This is not a criticism, but it's just the fact you have to optimize your mix for every RNA that you do, but you do it anyway. You optimize your magnesium, your concentration of all the NTPs. For the ligation, in our hands, we are at 200 micromolar samples. That would be great to even increase this further, but this is not trivial.
Wonderful. Thank you so much, Harald, for a wonderful presentation and for answering all of the questions that we had come in.